Hold on.
Hi, Jim.
Hey, how are you doing?
I'm doing good.
Yeah, I see you're at home. I didn't get to go home today. I'm staying here for this great meeting or podcast we're doing. So I'm at work right now. But I'll get to go home and have dinner soon. Don't worry. Won't be long. Won't be long. Won't be long.
Just hang on for another hour and 20 minutes.
Is that what it is? Okay.
Well, yeah, we don't actually start the webinar until 7. So
Oh, okay. Yeah.
I'm
And,
going to turn off the light behind me. Hi, Rachel.
Okay. Yeah. No problem. Oh, so you have people already here. Looks like.
Well, Rachel and Diana should be on soon.
Rachel is here.
Yep.
Hi, Rachel.
Hello. You're recording right now.
Yeah,
I am. I'm recording.
I
Yes.
am. When I turned it on, it went to record. So.
You know, you can pause that. You can pause that.
Yeah. Barry, would you like to pause that?
Yeah. How do we do that?
I think it's up at the top. I think it'll say pause at the top if you put your cursor up there. Try that. Did it work or not? I can't pause it.
Oh, okay. Hold on.
You have control over it.
Yeah. It's saying recording, but.
It should pause though. There's a button that says pause and you could.
Hi, everybody. Thanks for joining in. This is our webinar series on how to help landscapers develop eco-friendly programs to take care of people's properties, both turf and woody ornamentals. One of the things that got me started in changing the way I looked at landscape maintenance was learning about the soil food web. And it really changed my whole perspective on everything I do in the industry. And just recently, I connected with Dr. White, who is a professor of, excuse me, plant pathology at Rutgers. I looked on his website, and he has so many papers he's written, it's ridiculous. So anyway, I'm just going to momentarily pass this over to Jim. He'll give his talk. You can put your questions up and that will be taken care of. And then at the end, we'll just do a little recap. So Jim, thank you so much. It's really an honor to have you on, and I can't wait to hear what you have to say. The floor is yours.
Thank you so much, Barry. I'm really glad you guys invited me. I'm excited to talk about this stuff. And of course, the number of papers, of course, you know, that's my job, right? So I have to, they make us write papers, but of course, you know, we wouldn't be here doing it if we didn't enjoy it. But anyway, and some of those papers are on some really interesting things. You know, nobody was really interested in what I was working on for most of my career, you know, until 2018. And we published a paper that attracted a lot of attention on the rhizophage cycle. And then all of a sudden, you know, people were interested. And so anyways, that's part of what I'm going to talk about today. And the other thing that I'm going to talk about is nitrogen fixation by bacteria in plants. And that also is something important that we're working on and that people have not really studied very much. And in fact, plants are full of microbes and some of them are fixing nitrogen, and I'll talk about that. That's what we're studying. One of the things we're studying. So anyways, the tidal rhizophage cycle, and that's what we call this process of in roots, and then nitrogen fixation, which you're aware of. So what is an endophyte? All my career, I worked on endophytes. Endo meaning inside, phyte meaning plant. Basically, what we're talking about are microbes that go inside plants. And working with turf, you're going to be familiar with the grass endophytes, and these are actually fungi that go inside cool season grasses, and they make those grasses resistant to diseases and increase stress resistance and so forth. The picture here to the left actually shows one of these endophytic hyphae. So this fungus, in this case, is an endophyte in the grass. This blue line and these are now inside leaves. So this particular one is in a fescue grass, and that's what you're seeing here. Ornamental, but also turf and so forth. But actually, all plants have endophytes, and it's not just certain grasses, as you would think from the fungal endophytes in grasses, but any kind of plant you can think of has endophytes in it. And these endophytes are really, really interesting because they will actually go inside the plant cells themselves. So they'll enter the cells of plants and be inside the cells with the cell cytoplasm. And I'll show you that. They go into the roots, they'll go into root hairs, and I'll show you that, they'll go into leaves and so forth. So they're all over plants. They're bacteria and they're fungi. Okay, so here's an example. This is considered this plant. This is from the desert island of Bonaire in the Dutch Antilles. And a number of years ago, 10, 15 years ago, I did some work in Bonaire, great place to do field work. But this is a cactus that occurs all over this desert island. And way up at the top of the plant, they have these fruits. And if you look at the fruit, just like prickly pear fruits, take that, get the seeds out, and then germinate that under a clean environment, and you get a little seedling like this. And this one actually is stained. This orange stain is a stain that will enable us to see the bacteria inside these roots. This is the root hair. The root hairs of those several hairs crossing the screen of that little cactus. And you see the little dots inside that hair. Those dots in those hairs are actually bacteria. Those roots become filled with bacteria, and you can visualize them if you stain them. And this you have to have stains. You can't find them, you can't see them if you don't stain them. But we've been working for, as my career length, 40 some odd years working on endophytes, and so we figured out how to stain them. And so this actually is one of those hairs, and you can see the little blue dots in there. Those are those bacterial endophytes again. So those bacteria, endophytes, all plants have endophytes, and actually they have a community of microbes that go into the plant. So every plant contains a multitude of microbes go in there, and they don't cause disease. These endophytes are not pathogens, they're non-disease agents. So that's one important thing. You got to have a microscope to see them. Crop plants, consider tomato. You think of tomato as a pretty much, tomato, we think, well, there's no bacteria, anything in these tomato plants, except for what might be on the surface. So if you wash it, it's going to be fine. But in fact, that's not true. In fact, if you look around the seeds, in the cells associated with the seeds, there's going to be bacteria, multiple species of bacteria. And if you consider inside, if you look inside these tomato seeds or near the embryo, there's going to be bacteria as well. So these even healthy plants host bacteria. And various species, they're not just one. These are bacteria that can come from the soil too, although these plants would suck these microbes out of the soil. This is actually one of the, and that's where these microbes come from. They essentially come from the soil initially. The plants suck them out of the soil. But this shows one of the bacteria, the endophytes, that's in tomato. And this is one we isolated out. This is one called Micrococcus luteus. We like it because it has its cells in these little clusters of 4. Tetrads, little clusters of 4. We like that because when we put that in the plant, we could track it where it goes. We don't need to put any kind of special dye on it or anything like that. We can tell because nothing else in the plant has these tetrads. And so it's a experimental organism for us. This is tomato root. And you can actually see the root tip here to the right. And you can see the meristem, the root tip meristem there a little bit, just around where my arrow is going. And then you see this area, this kind of orange, purple. This is actually where bacteria are entering into these root cells. They enter into the root cells. So these roots, and I'll show you to the left here, look all the way to the left and you see the hairs. So these bacteria are entering into these cells. They're being processed by the plant, and then surviving bacteria are triggering root hair elongation. And if we remove those bacteria completely, what happens is roots don't form hairs anymore. So, I mean, and we can do that routinely. We can do that with any plant. It's really cool. In other words, the bacteria have to be there to have root hairs. This shows a weed. This is Rumex crispus. You're all probably familiar with this, but it's one of our favorite experimental plants because we can get the seeds and clean all the bacteria off of it. Then we can put whatever bacteria on it we want and see it goes in, see if it goes in. And this actually is an area of the root right around where the bacteria are going in near that tip. And you can see these cells here, big old cells, and you can see our micrococcus, the tetrad. See the little blue dots in there? That is that micrococcus inside that root cell, those root cells. Okay, so we can track it. This is where it goes in. And when it goes in, the bacterium will retain its wall. And so we can see this tetrad shape, the shape of this. Actually, what will happen after that is that the plant will then hit those microbes with reactive oxygen, with superoxide, and extract off all the walls and change the shape. The tetrad shapes will disappear. I'll show you more detail about how that works. But these microbes, every microbe that we get for the most part, especially in roots, is a soil microbe. Now, they may be better than standard soil microbes because the plant selects and kind of keeps the same microbes, but they all come from the soil. They're known to be soil microbes. Things like bacillus and pseudomonads and stuff like that. But the plant will take the best of these microbes and it will put it on the seeds and in the seeds. So that then when you have a seed produced from a plant, if you don't kill those microbes on the seed, or you don't treat it, or if you treat it properly to preserve those microbes, then when the seed germinates, the bacteria will then go into that seedling, and then it will be there to stimulate growth in those seedlings in the soil. So by putting the microbe on the seed, the mother plant is ensuring that the baby plant will have microbes there, and it's all seeds, as far as we know, do this, that we can find. They vector their microbes in seeds, but you can damage that. If you sterilize seeds or you pick seeds too early before the microbiome is able to mature on the surface of the seed or in the seed, or treat it badly, the microbes may not develop and seedlings won't develop properly. Well, this intracellular colonization or movement of bacteria into plant cells, roots specifically, was noticed by some investigators in 2010 and in Australia, in Queensland, Australia. And what they did is, this is one of these young lady here, Chani Pongfu. They actually wrote a paper that they published. They saw these bacteria going in and into root cells, and then they wrote a paper titled "Turning the Table: Plants Consume Microbes as a Source of Nutrients." And then they named this process rhizophagy. Rhizo for root, phagy for eating. And that denotes the idea that these roots are eating bacteria. So it's it's a little more, it's a little more sophisticated than that, a little more complicated. But that essentially is what's happening with many bacteria, is they're being used as food for plants. And this is from this paper, Turning the Table. And the picture, if you look down here at E, you can see a root hair. And you see the little green dots. Those actually are yeast cells. And of course, it's a yeast, it's a fungus, but yeasts will also do this. What they did is they labeled their yeast with this green fluorescent protein, essentially tagged it. That way, when they put it in the plant, they can put it under a microscope, a fluorescent microscope, and they can visualize those yeasts that they put in inside the plant. And what happened after a few days is these yeasts and the bacteria, they also used E. coli, they disappeared from within the plant. And so they considered that that's evidence of plants consuming these microbes. Well, we've figured out since 2010 that this process is actually not just as simple as that in fact, this appears to be a cycle that's happening. And what's happening, and the cycle, we called it the rhizophage cycle because we're respecting the scientists in Australia who described the process, discovered the process, and named it. So we varied that name just slightly to put the cycle there. But what's happening here in the rhizophage cycle is that plants, all plants, and their roots, at their root tips, they secrete exudates, and those are sugars and sometimes amino acids and sometimes organic acids. They secrete that into the soil, and that is an attractant for these microbes. And the microbes are attracted then to the root tip, and once they're attracted here, they're cultivated at this tip, and then they're internalized by the plant. They're taken in, they're absorbed into the plant. We don't exactly know how they're absorbed. There's some hypotheses, but it's a lot of uncertainty. How we just know that they get in because we can see them getting in. We can actually see them going in the process of going in. So we can see it all almost in real time, photographing it. But they go in, and then what happens is once they're inside the root cell itself will secrete superoxide onto the microbe. Superoxide is oxygen, basically. It's a very potent form of reactive oxygen, and it's potent. It will zap off. It zaps the cell walls. It oxidizes the cell walls off of those bacteria, and they form these naked protoplasts. And then the plant will continue to extract nitrogen. The root cells extract nitrogen from those microbes. Surviving microbes will trigger root hair formation. The way that they trigger hair formation is they're producing plant hormones, and we can measure ethylene and detect ethylene there, and also nitric oxide. Both of those are plant hormones, and both are related to elongation, root hair elongation. And so these microbes in those hairs cause elongation, and as these root hairs elongate, it ejects the bacteria out into the soil. So microbes exit the hairs, but by then the plant has extracted the nutrients already from those microbes by taking off the cell wall, and a lot of the nutrients are on the cell wall, but also by degrading some of the cells. The contents, things like potassium and calcium, get extracted from internally in those root cells. So once they're ejected from the tips of the hairs, they'll reform their cell walls out into the soil, and they'll also reform if they have flagella or any swimming structures. They'll reform those, and they'll go swimming back out into the soil later on, theoretically at least, to be then attracted once they acquire nutrients, to be attracted back to the tip of the soil. And like I say, we're trying to figure out exactly, questions like, how does the plant determine which microbes to take in? Because it does select. Certain microbes come in, certain microbes don't go in at all. So we don't fully understand that, but we're working on that. We're working on that.
Jim, I have a question for you.
Yeah.
This is really interesting to me because I've been learning about this for years and years, and I'm hearing something different from you. Where in the past I had always heard that the root hairs would send out nutrients like you have there on your slide, and that the microbes would also just exude nutrients and the plants would absorb the nutrients. But you're saying that the plants are actually absorbing the microbes too.
Yeah, the plants are absorbing the microbes. Now,
Good.
I want to make something clear at this point, and that is plants are doing everything that they can to get nutrients, right? Including swallowing bacteria and yeasts into their cells and oxidizing it or digesting them to get nutrients. But they're also absorbing solubilized or nutrients that are soluble in the soil solution, right? The hairs will go out and absorb those, and other cells actually will absorb those. They'll also get mycorrhizae. So they're doing multiple things in order to get nutrients. And if you consider like a legume that has rhizobia, well, they have nodules, and they're cultivating in those little nodules bacteria in order to get nitrogen. Okay, so they're doing multiple things, and rhizophage cycle is one of the things that we did not know that they were doing until very recently. And so this is their direct connection with these soil microbes. Like grasses, plants like grasses do rhizophage cycle a lot. This is a drawing of a corn root. This is the root, and this grasses have these branching root systems, as everybody here knows, and all those branch tips actually are where rhizophage cycle is happening. So at every one of those little tips in those branches, you get this rhizophage cycle happening, and plants are consuming microbes. And this is only happening at the root tips, by the way, only at the root tips. So if you don't have any root tips on a plant, or you have only a few root tips, it may not be doing this very much. It may be doing some other things to get nutrients. But okay, this actually shows, this is a grass. This is not a turf grass. This is one of the grasses that we work on. It's an invasive plant, and this is one called Phragmites, invasive Phragmites, australis, and it's a real problem in New Jersey. So we have a little bit of funding to work on that. So a lot of the work we've done has been focused on this plant, and everything else we do to kind of extend from there. But this shows a root, a branch root. And you could see actually these little blue clouds out here, those little blue dots in there. Those are the bacteria that the plant is cultivating at that root tip. So it's here with the exudates that are going out. The sugars and the proteins and the organic acids are going out to feed these microbes and control these microbes, feed them, attract them, and so forth. And what happens is the bacteria here are internalized into these cells right at this interface. They go into the cells where it's kind of dark blue. That's where the bacteria are entering, right there into those cells. When they go in, they look like this. And this is one of these Phragmites root epidermal cells. And the black arrows actually show these bacteria. What happens is the bacteria, once they're in, they're hit with superoxide, they lose their cell walls, and they form these spherical structures. And so you can see these round things. Those actually are these bacteria in here. And when they lose their cell walls, one thing that happens is, well, the plant gets nutrients from that, taking the cell walls, but the microbes are more susceptible to oxidative damage, to being zapped by that superoxide. And so you see, if you look at this lower arrow, you can see there's a little cell at the bottom that stains blue, and then you see the bigger cell up here. That's an older cell. So that's the little one is younger. What's happening is the younger one is staining blue because it has protein in it. We used a protein stain called aniline blue that stains protein. And so you can see the proteins are there, but these older ones are being zapped with superoxide, and they're losing protein. They're not staining blue. They're also simultaneously in these cells, you have the protein actually in these bacterial cells is linked up with cations like calcium and mostly like potassium, a lot of potassium in there. And so they're getting potassium by zapping the proteins in there in those cells, those protoplasts, and making them leak potassium. Then they can absorb that. But this is a really cool process. This is actually one of my graduate students, Celeste Zahn, And she did, we had to get in like the Australian group, we had to do our own tracking. So what she did is she took a bacterium, a Pseudomonad, she labeled it with this M. cherry, which is a nuclear stain that when you visualize it under fluorescent microscopy, it stains this red color. And that's the bacteria basically. That you put it on the plant, and now she scanned it under the microscope, and you can see all those red dots. Those are all her bacteria that she put in there where they went into the cells. So those are the root cap cells that came off. But you see the same thing in the other cells of the root. These bacteria, they're not actually going into the cytoplasm of the root cell. This is a diagram of a root cell, plant root cell. And the gray is the root cell wall. And what's happening is the bacteria are going through that cell wall, and they're going into the space between the protoplasm, or between the cytoplasm and the membrane of that root cell. The little blue line is the membrane of that root cell, and then the cell wall. And so they're stuck in that area called the periplasmic space. So they're just underneath the cell wall. And it's in that space that the plant puts that superoxide. So this 2O minus is the superoxide that the plant produces. The plant can detect the microbe and hit it with superoxide. The way it detects these microbes actually appears to be because these microbes are producing ethylene, so that the plant responds to that ethylene with this superoxide. That's the first interaction. But there's something else happening here that's really kind of interesting. And that is that the plant cell is moving these microbes around in these roots. It is circulating these microbes. We think that's important because that enables the plant to kind of break down these gradients. If nutrients are flowing from the bacterium to the plant, if it's staying there, there may be a gradient, a concentration gradient that builds up and then slows flow down from the bacterium into the plant, and the other way too. But because it's moving, the gradients break down, and nutrients will flow very fast between the bacterium and the plant, and then the other way, because it is a kind of a symbiosis, and there's a nutrient exchange that is happening here. But this is cyclosis, by the way. I'll show you this in root hairs. You've never saw it, you never thought, I'm sure few people have ever seen it. Even scientists have ever seen it. Go ahead.
We have a few questions in the chat. Is this a good time to
Of course.
ask? Okay, fantastic. John would like to know, does the term Endomycorrhizae by extension refer to bacteria inside the mycorrhiza?
That's a good question. No, it does not. There are endohyphal bacteria, and so that's kind of what you're thinking of, endohyphal bacteria. There are those. We can see that, and other people have published on that, so we know that's happening. But Endomycorrhizae actually refers to a group of mycorrhizae called the VA, vesicular arbuscular mycorrhizae, or sometimes just called the arbuscular mycorrhizae. These are mycorrhizal fungi. They're fungi that will go in their soil, fungi, but they don't live freely in the soil. They live in association with plants. And what will happen is they'll grow, put a hypha into a plant, and then they'll start getting nutrients from that plant and bringing soil nutrients back to the plant. And so the Endomycorrhizae is a little different, but that's distinguished from the Ectomycorrhizae, which is also fungi that go on the surface of the plant. So these are things that plants can also do to get nutrients. So you just add the mycorrhizae, the Endomycorrhizae in, which you have in grasses, and this rhizophagy cycle, and you've got primarily the two main ways that grasses use to get nutrients from the soil, other than where they're absorbing dissolved nutrients from soil solution. So Endomycorrhizae is one way, another way that they get nutrients. There's another question.
There was a
Right.
second part, well, there's several. So there's a second part to that question. So he
Yeah.
asks as well, is that bacteria contiguous with the endophytes in the plant itself? And I'm,
It's a, yeah, really good questions. And to some extent there is. Now, some bacteria will just do in the rhizophagy cycle. They'll just go and stay in the roots. But there are some bacteria that move from the soil, into the roots, and then move up into the rest of the plant. Now, the fungal endophyte that you know of in turf grasses doesn't do that. It stays in the leaves and goes in the seeds. But for these kind of endophytes, the plants can get them out of the soil and they can shuttle them up into leaves also. And you'll see, and then go into seeds and on seeds as well. So yeah, so that to that extent, they are contiguous, but not every single individual bacterium will do that. Just certain ones will go into the aerial parts. Okay, other questions?
okay, yeah. John continues to ask another question, and he's asking, can the exudates actually tell roots to uptake water and turn off that update? And I ask because there is a company making that claim for their drip tape product.
Say that question again. Say that question. Can the exudates turn off water?
Can the I'm guessing it's exudates. It's eudates, but I think he means exudates. Exudates actually tell roots to uptake water and turn off that update. Can the exudates actually tell roots to uptake water and turn off that update?
So I will tell you what I think. What happens is that exudates are signals and food essentially for soil microbes. So these plants are using their exudates to attract soil microbes and to manage soil microbes. What actually happens is that plants will internalize these bacteria that they attract to the plant. And then because this is an oxidative interaction with plants, plants then become more hardier, more stress tolerant. And that stress tolerance goes so far as making plants more resistant to stresses. There is also a phenomenon with regard to bacteria going into roots, and that is that what happens is plants will produce more exudates around the roots, and then that will remain moist in the soil. And so you can sometimes see when you pull these plants, grasses in particular, in very sandy places, you pull the grass roots out of the soil and you'll see like a column, a little rhizosheath, it's called, a little sheath, a little soil sheath that goes around the roots. And that has to do with the moisture that comes out of the roots around where the microbes are. The resistance to drought, or the ability to survive with less water, is basically stemming from this oxidative stress tolerance, making plants hardier. And so that's the best way I can answer that. There is a relation. Now, putting water on the plants, if you put microbes on the plants, yes, you can make these plants more resistant to drought. But putting water on the plants, well, you just give them water, there may be some additive. I know people do interesting things with nutrients. In some cases, people put sugars in soil, sugar water or something, and that cultivates microbes, releasing nutrients, and cultivates more of the microbes, so that you have more microbes than being attracted to roots. But you have to be careful with that, because if you put too much sugar in soil, the bacteria won't go to the roots because they'll be swamped out. The whole signal will be swamped out with all that sugar you put in the soil. So you have to be real careful with that.
There's a couple more questions. Kent
Yeah,
would
go ahead.
like to know, does the plant signal microbes such that the microbes provide nutrients needs at each stage of growth?
We're not at that level of understanding. Presumably, yes, but we're not at that level of understanding. What we do know is this, that plants, from experiments with corn, we know that plants that want to attract nitrogen, that need nitrogen, and therefore they want to attract nitrogen fixing bacteria, what they do actually is they will stop secreting amino acids that have nitrogen in them, right? So they'll stop secreting that, and that will make the microbes themselves, they'll get sugars out there. The plant will secrete sugars, but no nitrogen. And then those microbes have to fix the nitrogen, and then the plants can then internalize those. So the plants can select. We don't understand all the details of how they do that, but yes, they can select. Is it different stages of growth? That makes sense, but I can't answer at that level.
Okay, fair enough. Mark has a question. He would
Okay.
like to know, do you have any thoughts or knowledge on Art Johnson's from Penn work on southern Chile where fungal mass in the soil?
In Chile, the country, or Chile peppers? Chile peppers, or I don't know, is it Chile?
It looks like southern Chile, the country.
Chile, southern Chile. You know, I do not. Yeah, microbial mass makes a difference. Fungal mass will make a difference, but I'm not familiar with Art Johnson's work, and that's just because we probably don't move in the same circles, basically. That's the best I can do on that. Next question.
No, we're
Caught up.
Yep.
Okay, so how does the plant zap these microbes with superoxide? Well, they have in the root cell membrane, they have this enzyme called the NADPH oxidase. And this is in the root cell in the membrane. And what this enzyme does, it will take oxygen from the air and it will produce superoxide. And then that's the superoxide that it will hit those microbes with to oxidize off the cell walls. But this is atmospheric oxygen. So plant roots have to have access to oxygen. So if you deprive the plants of oxygen by, for example, flooding soils, then this whole process doesn't happen and plants can't get nutrients. And so you have very poor growth. There probably some other effects of removing the oxygen as well, because the cells have to breathe. But one of the effects is this failure to produce superoxide and do rhizophagy cycle. So this inhibits the rhizophagy cycle. We can also elevate carbon dioxide, get carbon dioxide, elevate it pretty high. And what that does, it will also inhibit the formation of superoxide. Without that then high levels of carbon dioxide mean there's no superoxide, and then the plants can't get nutrients out of their microbes. There's a lot of work that shows with the increasing, well, if you put too much carbon dioxide around plants, they actually will grow fast, they grow great, and they have lots of photosynthesis because that CO2 enables them to fix lots of sugars, but they start losing all other nutrients, calcium and nitrogen and other iron and all kinds of other nutrients, because those other nutrients are coming in this rhizophagy process. At least part of them are coming there. So that part, this vanishes. And so you have to be careful with pushing plants using elevated CO2. How much is enough? I don't know. In our experiments, we put a lot, but this is a known inhibitor of nutrient uptake, carbon dioxide. This is what the bacteria look like when they hit with superoxide and they lose their cell walls. They may have rods here when they have their cell walls. Then the superoxide, once they go in, superoxide then oxidizes off the cell walls, and they have these little spherical protoplast forms that are called L forms. Those have been named. We didn't invent those. They're known from medicine. They've been known for a long time. The L forms, the L stands for Lister. So at the Lister Institute, that's where they discovered these in medicine.
James?
Yeah.
Are any of the bacteria expelled in the vapors when the stoma are open?
No, no, they don't go out into the air. They will go into the leaves though, and they will go onto the leaves. And I'll describe that process. I'll show you that before very long. I'll show you that. So yeah, they do go up there. They will go onto the surface of the leaf. They don't go in the air, but you can get them spilling on the leaf surface. And I'll show you that, how that happens. But back to the roots and the rhizophage cycle, the microbes go into the roots. If you don't have those microbes there, roots don't develop properly. And one example is the gravitropic response. And mostly plants have roots that grow down into the soil. That's well-known gravitropic response. If we, especially in grasses, turf grasses, if we remove all those bacteria, then what we get is those roots no longer go down. They no longer go with gravity. Instead, they'll stay on the surface and just lie on the surface, or they'll go into the air, roots going into the air. And the other thing we see that's really interesting is without bacteria in the root hair, you don't have any root hair elongation. If you don't have bacteria, if we take our turf grass seeds and then we sterilize them for long periods of time to remove all the bacteria on them, the seedlings that come out of those seeds will make roots, but the roots will not have any root hairs. So they can't form root hairs without those bacteria being in there. So we have always thought that these root hairs are just for absorbing, but it appears that, in addition to absorption, because they definitely may be doing absorption, they are also moving these bacteria out into the soil. They're ejecting bacteria, and I'll show you that. This actually shows, this is a warm season grass. This is a weed for us in the Northeast. This is Bermuda grass. This is a weed here, but in the southern US, it's a turf grass. This is actually Bermuda grass, where we removed the bacteria from the seed and then we germinated the seedling. And you can see the root tip here to the left, and you see a little bit older part of the root here. And you notice there's no hairs there. But when we put the bacteria back on, remove the microbes from the seed, then we put bacteria back on, you get now hairs forming immediately. This is from the same experiment. You can see those hairs. Those bacteria are necessary for hair formation. This is older part of the root. You can see the root axis here. You see the hairs coming out, little dots. Those little brown dots are bacteria in those hairs. The hairs are full of bacteria. And those are the pseudomonads that we stained. If you look at the cells here, you see the brown dots on the root cells. The root cells are full of bacteria. So you got bacteria all in these tissues. These roots really are eating, especially grass roots. They're eating, they're really gorging themselves on these bacteria, like pseudomonads and like bacillus. Some of the biostimulant microbes that you can buy go into the rhizophage cycle, some of the bacillus biostimulants, and you'll go into this process as well. This is a root hair. You can see the little brown things in there. Those are the bacteria. Those are the bacteria. These plants can gorge themselves on these microbes. They become filled with microbes.
James, Steve would like to know, how is grass seed endophyte enhanced, and is it temporary?
No, well, it could be temporary. So with the cool season grasses like fescue grass and ryegrass and some of the fine fescues, hard fescue and so forth, they have these fungal endophytes in them. A fungal endophyte that's actually in the genus Epicoi. We just call them in the turf industry, we just call them fungal endophytes, but they're actually a specific kind of endophyte. And they go only through the seed, and they go into the leaves of the plant and into the seed of the plant. Endophyte enhanced means that the breeders, when they bred that variety, they selected it to have the fungal endophyte in it. But a lot of these other endophytes can also be there. A good healthy seed will have bacterial endophytes on them. You just want to be careful not to to hurt those. They'll be naturally there, or you could even add them too. But okay, so anyways, did that answer? I think that answered that pretty good. Endophyte enhanced means the breeder put it in. Now it can be temporary, because what happens if you store seeds improperly, maybe you put it in a barn or something for 6 months to a year, it gets hot over the summer or something like that, that fungus will die in that seed. And so that is a temporary, and then there's nothing you can do to get it back. You've got to go buy new seeds. So when they say Endophyte enhanced, it's a specific kind of fungus that they're using, that the only way they can get in there is the breeder has to breed it into the seed. But you can lose it, it can die. Yeah, we've done a lot of that work. So the question here is, with these bacterial endophytes, is why do they cause root hair elongation? Why does the bacterium have to be in that root in order to make that root hair elongate? The answer is, turns out, done by my student here, Ivy Chang. The answer is that these bacteria are producing plant hormones. And this just shows the tip, root hair tip. See all the bacteria in the tip? Well, those bacteria in that hair tip, all that brown stuff, they're also producing ethylene, which is a plant hormone, which is a growth hormone in plants. And that ethylene will then cause this root cell to grow. And as it grows, then these bacteria will be replicated. But some of these bacteria, as soon as you have a growth spurt, it'll eject some of those bacteria out of those hairs. So the bacteria is part of the rhizophagy cycle, that the hair grows, the bacteria get ejected. I'll show you that. Oh, and I should show you this. This is really interesting. This is, I talked about the bacteria in the root hairs. Well, they're not just sitting in there static. In fact, they're moving. The plant is moving them. This is a plant I'll show you about. This is a sedge. You can see this in the grasses too, fescue. You see this in fescue and other plants. But the sedge is really nice. This is a sedge growing on rocks. It doesn't have any soil, but it does have lots of bacteria. And this actually shows one of its hairs below through the microscope. And you can see the little brown areas. Those are where the bacteria are in this periplasm, in the space around this, in this root cell, right? It's just underneath the cell wall. Everywhere the red there's bacteria in there. And that's fixed, that's stained and fixed, it's not moving. But here is a living root hair. And this is photographed through the microscope, grown on a Petri dish, a plastic Petri dish where you can see the bottom, right? See the bottom, see through it. And so we can then grow it in this agarose on this Petri dish and turn it over and look at it alive. And this is what you never see in the soil, in your turf, when you're looking at it. You have the hair, but you see now this movement. These are the clouds. See these little shadows moving around. These shadows are the bacteria that you see down here. The plant is moving these microbes around and around in there. The plant is processing these bacteria. The plant is like a machine in absorbing these microbes, extracting nutrients from them, and then processing them to further extract nutrients from those microbes. And that's what you're seeing here, this movement. This we call cyclosis, or we're calling it more periplasmic streaming, because these microbes are in the periplasm just underneath the cell wall. But it's cyclosis or streaming that's happening. But this movement also makes those microbes replicate, these protoplasts, and you can see them here, these clusters of them. These clusters, each cluster of bacteria here probably started from a single bacterial cell that was replicated in the plant. And so the plant is actually replicating these microbes. And so a few microbes will go in at the cell at the root tip, and then the plant itself inside the root hair will clone those microbes. And those microbes that get cloned are probably the ones that are benefiting the plant the most. There's a lot we don't know about it, but that's likely what's happening. And in fact, we think they're fixing nitrogen inside these roots even, and transferring that. We published a paper on that a few months ago. But this shows one of the,
James, John has a question, and he'd like to know, will endophytes enrich soils that are deficient in organic matter and or other nutrients?
They will.
clarifies by saying, what I'm asking is, will the bacteria replication enhance the soil's fertility?
Yes, the plant, yes, it will. Well, what's happening is the plant will be increasing, will be getting nitrogen from these bacteria that it's taken in. And these bacteria that survive this process are going to be the ones that are ejected back into the soil. So the plant is actually going to be enriching the soil microbial community with these microbes that it cultivates and ejects from the, so they are, so the plant itself will be enriching the soil. So that's a good point, a good question and a good point. Yeah. Did I cover that good enough?
I think so.
Oh, good. Thank you. Yeah. Yeah. So this shows a root hair, and you can see this streaming happening. This is about as fast as you actually see it. I mean, this may be double speed, something like that, maybe triple speed. But it's, when you look in the microscope, we see it moving about almost this fast. And we measure this movement of bacteria in these hairs at a rate of 8 to 11 micrometers per second in the sedge, but we measured it similarly in tall fescue at about the same rate, similar rate. So in the root hairs of tall fescue, so they're really moving these things around. And notice they're accumulating at the tip. You can see these little white ghost things. Those are the bacterial protoplastic accumulating at the tip. These bacteria in the tip will secrete the hormone, the ethylene, and nitric oxide will be formed as well. And then that will cause a growth spurt, which will then cause some of these bacteria to go back into the soil. And I'll show you a picture of that so you see how that's going. This will happen repeatedly. A growth spurt, ejection, a growth spurt, ejection, and so forth. And each time the plant will then rebuild the populations in the hair until you get a bunch more here at the tip, and then it will grow and eject, and then build up again, and then eject, and so forth like that. It's really, really a cool process. This shows one of these sedge hairs. You see how fat it is. These are the bacteria outside. You can see some of these look like rods. They're reforming their shapes. But inside this root hair, you can see some of these are stained, but some aren't. There's white. There's a bunch of them in here. These are full of bacteria, even though you can't see the individual bacteria here. They're fat. These hairs become fat with bacteria. If we were to remove all these bacteria, these root hairs become skinny. There's actually in the middle of this a little root hair cell that's a skinny little needle-shaped thing in the middle of that hair. So most of what you see around this, making this hair fat, are bacteria. This just a little diagram, a little cartoon that shows that ejection process. You can see what I was talking about. They're in root hair, they accumulate at the tip. They produce hormones, growth hormones, ethylene that causes the hair to grow, a growth spurt. And then these are ejected out of little holes. And then the remaining ones from the side then are taken back in, and then they're replicated. They replicate, they replicate, and they form a bunch that accumulates in the tip again. Now you say, why are they accumulating in the tip? Well, because that hair tip is very elastic. It's very, you saw how fat it was in the previous image. Well, these are soft. These are really soft walls. They're elastic. So the tip is soft. And so it will expand. And so when these bacteria go there, they essentially expand and they don't get caught. They accumulate there rather than being moved back in. This is tomato. And it's the root hairs of tomato. And you can see the microbes are actually being ejected from this. When you look at 3, if you look down here at 3, I don't know if you can see my little hand down there, but there are 3 little holes, little pores in that tip, where the tip of the hair, where these bacteria are coming through, and it squirts them out. And then these bacteria that squirted out as protoplasts, but then these bacteria will then reform their cell walls and any flagella, and then swim out back into the soil. So, and here's another little thing that tells you what the plant really is in charge of this process, because when the plant secretes these bacteria out, it also will secrete a little bit of carbohydrate out with it, a little bit of sugars out with it so that these bacteria will have some energy in order to rebuild their cell walls. And don't forget, for a plant doing photosynthesis, sugar is cheap. I mean, it's really cheap. In fact, this whole process that plants are using with plant exudates, putting these nutrients into the soil, it's been estimated it's as much as 40% of the sugars that plants are producing in photosynthesis. So they're sending almost half of their nutrients out into the soil to cultivate these microbes. So, I mean, this is an important process for plants. It's surprising people haven't seen this before. Now, nutrient absorption. We've done several experiments that show, and others have also done some experiments that show that it's a nutrient absorption phenomenon. This is one of the people in my lab, Shuai Zhao, who came and he worked on winter wheat, another grass. And what he did was he isolated some bacteria from winter wheat seeds, some bacillus bacteria, bacillus species from the seeds. These are seed vectored bacteria. And then he sterilized to seeds, removed all the bacteria, and then he put them on one at a time. And then he grew the wheat with and without the bacteria, and then he measured the nutrients that went into the wheat leaves. And this is from his experiment, and this actually shows us winter wheat with no bacteria added. So this is our no bacterial control. And without bacteria, you see the plants grow, put them in the soil and they grow. It's not like we killed them, but you notice the roots are not growing. The roots are very short, and also the plants themselves are restricted in height. And these were grown in sterile potting mix. So this is a soil-like environment where they had other nutrients. So they could grow soluble nutrients, but nothing else, no microbes there. To the right is one of the microbes, one of the bacillus species put back on. And you see how big the roots are now. Same magnification, same experiment, same time, everything the same, but you see the roots are like 3 or 4 times the size. The leaves are even bigger there. And these are the same magnification. If you look at the tape below, you can see they're the same size tape. That's our standard laboratory tape. So we didn't like blow the one to the right up so it made you think it's big. This is the actual size. We're not playing any tricks on you. Here's nitrogen. This is the measured nitrogen. And here's the no bacterial control. You can see these plants got this much nitrogen. But when you add the bacillus in, you add the bacteria in, you see they got like 30% more nitrogen. And all 3 of those, you had 3 different bacillus, they're all doing the same thing. Look at phosphorus. With phosphorus, you can see no bacteria, you get so much phosphorus, but then you get about 20% more phosphorus when you put your bacteria on. Now, all 3 of those bacteria seem to be the same. But now with potassium, it's a little different. Look at potassium. With the no bacteria, the plants suffer tremendously from potassium. They don't have much potassium. Then he adds it in, adds his first bacillus, this first bacterium, and you can see it's like 3 times the amount of potassium. And with these others, they're not as good, but you got about twice the amount of potassium. So potassium seems to be a really important nutrient for rhizophage's cycle, right? For getting, and of course that's coming, potassium is coming right out of the cytoplasm of these bacteria because it is a solute. It is a potassium is inside these cells, providing the solute, providing the cations in those cells. So the plant is just, it's robbing the potassium from those cells, from those bacteria. So I did mention that these bacteria go into the leaves of plants, and the plants are putting them in the leaves. And this is the newest work that we've done, and we've just now gotten the paper that we're finalizing for publication. It's already been reviewed and accepted, but we're still making some revisions on it. But it's been minor revisions were proposed. So, but anyways, this is a paper that's going to be coming out pretty soon. And hosta, consider hosta. It's another monocot like grasses, and grasses do the same thing almost that hosta will do. These have endophytes that go into the leaves. So the plant is putting these microbes into the leaves and then causing them to fix nitrogen in those leaves. And I'll show you how that's happening. But to understand that, I'm not going to go into many details here because it's complicated. But there are, you know, some of you have chemistry, and it's been a long time since I've had chemistry. But chemical interaction, this is one of the chemical interactions that we have figured out. Basically, it involves the ethylene produced by the bacteria and superoxide produced by the plant. That causes those two will combine and we'll get hydrogen peroxide. And so we can measure hydrogen peroxide around these bacteria. We can stain for hydrogen peroxide and see it there. But this is the most important interaction, is this one. What's happening with the bacteria is they also are producing nitric oxide or antioxidant forms of nitrogen. This is protective nitrogen. This is nitrogen that the bacteria are fixing, and then they are secreting it in order to counteract the superoxide, to protect themselves from superoxide. And what happens is the superoxide produced by the plant combines with that nitrogen, either ammonia or nitric oxide, and maybe some other antioxidants there, but this is the reaction that we're seeing. Those are combining, and then it's forming nitrate. And the nitrate then is being absorbed into the plant cells, into the leaf cells, that have these bacteria in them. So they're absorbing this nitrate. And notice it's nitrate that's happening in this natural process. That's the nutrient that plants are absorbing from bacteria. So we think this is the reason that people are able to use nitrate as a fertilizer, because this is the process. This is the nutrient that plants are naturally getting from bacteria, the nitrate. Okay, the only problem is that if you give the plants nitrate, the plants don't need to do any of this stuff with bacteria. In fact, what they do is they stop. You give them ammonia or you give them nitrate, plants will stop cultivating bacteria, and the bacteria will stop fixing nitrogen. So you essentially stop by putting nitrate on the plant rather than letting the plants extract nitrogen from the bacteria. By putting that nitrate there, you cause the plant to stop doing this whole process. And that weakens plants because it makes plants less oxidative stress tolerant. Because they're not doing oxidative interactions with plants using this superoxide, they're weaker plants. Put nitrogen on, they don't do this process, then they're weaker. They are more susceptible to disease, they're more susceptible to stress and so forth. Oxidative stress, all stress is oxidative essentially. They're more salt tolerant, they can't tolerate mowing as much. Now, I don't know if you see that or not, but in theory, this is what's happening. And there's research that suggests that you make plants weaker, and certainly they stop all nitrogen fixing and they stop all absorption of microbes. They actually stop secreting exudates to attract microbes if you put nitrogen on them, chemical nitrogen. Now, sometimes we have to, but still, it would be better to find some other way to use the microbes in order to feed your plants nitrogen. So we did a big study of a lot of plants, like 33 different plants, looking at leaves to figure out where they're doing nitrogen fixation. And we did 33 plants. It's a table that shows that. And this is one of them. This is hosta. I think, let me cover, it's covered. I think this is hosta. And this is the epidermis of hosta. And this is actually a symbiosis that's occurring in the cells, the non-photosynthetic cells. So it's the cells without chloroplasts, like the epidermis cells, and hosta has these green areas, these variegations in them, these areas where it's not green, but they have green areas where they have chloroplasts. And then they have more yellow areas or whiter areas, lighter areas where there's no chloroplast. It turns out in the cells, in the areas where they don't have chloroplasts, you can find this. This is a cell from a hosta in that one of those areas, and you can see the nucleus in here. That nucleus has little dots in it. Those are bacteria. What happens is these bacteria move into the nucleus, and the plant cultivates them in there. We actually know they put sugar in there so that the microbes fix nitrogen, they replicate and they fix nitrogen in there, and then the bacteria are released into the cytoplasm. And so you see these spherical things, this actually are bacteria in vesicles coming out, the bacteria being released. And it's stained blue for nitrate. This blue stain is nitrate. And so what you're seeing here is that in the nucleus, you don't see a lot of blue for nitrate being produced, but as soon as it goes out, now you see the blue. You see the nitrate being formed, and these go out in the cytoplasm and they're circulated, and that nitrate is then extracted from them. To the right, you can see another host cell, and you can see this is actually showing ethylene, I think, ethylene, the blue there. And you see there's no ethylene while it's in the nucleus. That's a lot of sugar there, so they can grow and fix nitrogen. But when they're released, now the bacteria start releasing ethylene in order to get nutrients from the plant. And when they do that, that's when the plant starts putting superoxide on them, and they start releasing nitrogen, pumping out nitrogen. So that's a part of that process.
James, John has a question, real quick question.
Yeah, John, yeah.
And by the way, just a time check, it's 8:04. John wants to know, is chemical interaction number 2 stunted by applying chemical nitrogen in fertilizers?
Yes. Yes.
Short and sweet.
Yes, yes, it is. You see stoppage in various places. You see a stoppage, chemical fertilizer, nitrogen at least. I can't tell you about phosphorus and potassium. I don't think we see the same thing happening. But with nitrogen, you see plants, they stop taking in bacteria. So that's stopped, right? And then the bacteria themselves that are already inside the plant don't fix nitrogen. And they don't fix nitrogen because there's abundant nitrogen in the plant, right? So they don't have to fix nitrogen. So the whole process kind of is stopped. So yes, that second equation, the whole process, the whole rhizophage process is affected. This actually, look at this. This is I know, I'll let you say something just a second. Hold on just a second. Here's the nucleus. This is a living nucleus. And you can see the bacteria coming out of the nucleus. And this is stained for superoxide. So you can see it's being exposed to superoxide. As soon as it comes out, it's being hit with superoxide. As soon as it comes out of the nucleus. So you could see it real time through a microscope. We can show that. And these nuclei just get filled with these bacteria. It's a case. Now, what about the DNA in there? Well, DNA, we don't know whether the DNA is working in these epidermal cells or in these cells where we have this nitrogen fixation happening. We don't know about at some point, maybe these nuclei aren't needed for DNA after the plant cell is formed. I don't know, but the bacteria definitely go in there. Now there's I think there was another.
Yeah, so the point I want to make, you were quite clear on it. I was thinking it as you were talking earlier. If I'm not mistaken, what you're saying is that adding urea and synthetic nitrogen, while it's going to green grass up, it's very detrimental to the health of the plant, and therefore you might have more diseases and more insect problems.
Yes.
Okay.
Yes, that's exactly what I'm saying. Now, the truth is, I mean, sometimes, what do we do to grow plants? I mean, we do what we have to do, right? If you're having if you have soil that has only sand, you've got to rebuild your soil. You got to regenerate your soil, right? You got to do stuff to do it, and it takes time. So, sometimes we don't have a choice, but
yeah, but most times we do.
Yeah, you can make it. You can make it happen.
Yeah. I mean, in the turf industry, there's somebody going out to their turf several times a year, and in my viewpoint, there's no reason why they have to be putting down so much urea, and they could be putting down more organic matter.
Right. Right. Exactly. And that's the point. And that's the point of this work. I mean, this is the natural process that plants use. And it's logical to me that what we want to do is actually, if at all possible, support the natural process in plants for getting nutrients, right? As opposed to just throwing the nutrients on the plant. So if we know this is happening in plants, then if we can then support this by providing, say, organic forms of nitrogen that then the microbes can get and carry back to the plant, that would be better than putting those nutrients directly on the plant. So yeah, so here's another tree of heaven. This is another, we like to work on weeds. You know, I mean, that's why I'm in the turf, one of the guys in the turf center, one of the people in the turf center, but I always get in trouble because I love to study weeds. So we have a turf weed scientist here that I've been collaborating with a lot lately. But this is one of the weeds. It's tree of heaven. You're familiar with it likely, but if you look at the tree of heaven leaves, look at the leaves are covered with these trichomes. And those trichomes are full of bacteria, these filamentous trichomes. You can see the bacteria coming out of holes in these trichomes. So some of these bacteria coming out of holes are going onto the surfaces of leaves. And so we can see them piling up under these trichomes. So these trichomes are working, fixing nitrogen in here, inside the trichomes. The bacteria go in here, and now you have the interactions happening in here, but there are these holes here, and some of the bacteria fall out of these holes. It's really interesting. Here's another one. You know, it's cannabis is legal in New Jersey, but it's still hard to work on because it's not fully legal. And so one of my students is trying to work on it really hard when it's still illegal, when it's not fully legal yet. But what she did is she found that the trichomes on cannabis, hemp, leaves covered like this and the flowers covered like this, this is the trichome itself. You see it's got a stalk and it's got a head. Well, these heads, notice they're not green. They have bacteria in them, and they also have chemicals, but they have bacteria and some plant cells, but they don't have any chlorophyll. They don't have any chloroplasts. And that's important because chloroplasts in these same cells where we're getting nitrogen fixation would be doing photosynthesis. I mean, chloroplasts do photosynthesis, and that releases oxygen, and oxygen inhibits nitrogen fixation. So it's important that these cells, these trichomes where we're going to have nitrogen fixation, don't have photosynthesis, don't have chlorophyll, don't have chloroplasts, and therefore they don't get oxygen. Instead, what you see is the stalk is green, and that's where photosynthesis happens, and that's where the sugars are made that then go into the head to feed these bacteria and nitrogen fixation in the head. And I'll show you the bacteria there. Really cool. This is one of these heads. You see the red arrows, there's the bacteria beginning. This is early in development. You can see the little rods in here. The bacteria here, they're hit with superoxide. They're starting to, the plant is removing with the superoxide, it's removing the cell walls off, and it's making these protoplasts just the same way we did in the roots. And you see now, this is a bit older. You can see that trichome. You can see the 8 cells in the middle. Those are the plant cells. And you can see around it, the bacteria. These are the bacterial protoplasts. You see the interface of the bacterial layer and the plant. You can see the superoxide there. That's superoxide, the purple there, where the plant is putting superoxide onto the microbes. These are fantastic machines. We think, actually, this is the model of how these glandular, we think glandular trichomes are really like you see in cannabis. We think these are like the really efficient nitrogen fixers, but a lot of the chemicals, the cannabinoids, the terpenoids that you see in cannabis, in those trichomes, those also are reported to be antioxidants. And so what we think for a lot of these chemicals that form in the cannabis is that they scavenge oxygen around the microbes so that they reduce that oxygen inhibition of nitrogen fixation. So those bacteria in those glandular trichomes fix nitrogen very efficiently without oxygen around there because of those chemicals. Of course, the side effect is animals. That's our hypothesis, that the side effect on animals is the side effect, that the main effect is this antioxidant effect to make these plants fix nitrogen. Here's hops. Again, my student is working on hops, and you see these all over these cones to make beer, you know, for hops. The trichomes have all those chemicals in them we like, the terpenoids that are in hops, in beer. This is a developing one, and you can see the blue, purple here. Those are actually bacteria. This is bacteria in these developing trichomes. And here's a mature one. This is a fully developed on one of those scale cone leaves, one of those, we call them bracts, but they're leaf structures, right? and the seeds would form there, but this is the trichome, and this is stained for nitrate. And you can see how much nitrate, how blue it is compared to these other cells on that leaf. So those are really, really efficient at fixing nitrogen. Those are the most efficient. So I think I'm out of time, but basically plants are absorbing, all kinds of plants, all plants absorb these microbes into their roots. They get nitrogen nutrients from them, all kinds of soil nutrients. They use them to carry soil nutrients to the roots. They move them all through the plant into the leaves and into the flowers. And then they get nitrogen out of those microbes in the leaves and the flowers. So you get all kinds of nutrients in the roots, and then you get nitrogen happening when the certain ones are moved into the leaves and flowers. Here are some papers that you could find. These are all open access, and you can get to these papers online for free. And they describe some of the work. We haven't, like I said, the last one on trichomes, on nitrogen fixation in trichomes and leaves, we're just now getting accepted, final stages now for sending it back with our edits. But it should be out in another couple of weeks, something like that. We have some stuff they have to do, but it'll be out pretty soon too. This is not a one-man show. All these people contributed. Like, they may not be as bold as I am or as experienced as I am, but they put a lot into this. And they all contributed parts to this work, and they all deserve credit. Graduate students and postdocs and collaborators and so forth. And that is it. And let me see if I can figure out how to did it. Okay.
John, thank you so much. That was absolutely fascinating. The depth of what you presented is just tremendous, and it reinforces all the things I've learned over the years. Just a couple of questions I think we could help us out with. How do you think the best way to explain to turf managers that they can be much more successful by growing healthier plants than with applying all the pesticides that are trying to recover from poor practices?
Yeah. Personally, I mean, this is like my personal mission to inform the world what we're seeing in plants, right? And I think that once anybody sees the whole story, it changes their perspective. I mean, this is what we're seeing is, it's not complicated. This is not complicated molecular biology. This is, this is ecology that we can visualize with the microscope and see and discuss how it correlates with other observations that people have made and other data. And if somebody sees this and they start, and I think they start, well, it's perspective changing. It changes the way people see plants, and it will, and I believe it's going to, in the long run, change how we cultivate crops. And I think that in time, we're going to all develop. I mean, right now, there's a lot of people doing regenerative and kind of biological approaches to agriculture, but I think in time that's going to be mainstream, and it's going to be rarer to see people who are just putting, chemical fertilizers on plants, just the way we do now, right? That's going to change, I'm sure.
Yeah. And what I've been seeing also is that I don't know how much of the change has actually come from the industry itself, but there's a real growing demand for these kind of services and getting away from the chemicals. And that's what's really going to drive the change in our industry.
And I think people, people are all very smart, and once they see the whole picture, they start to, reevaluate. Things start going, and they start putting things together. And I think and I think in time, we're going to see a sea change in toward a more regenerative biological agriculture approach.
Yeah, I agree. And the information you're putting out there is extremely helpful. But what makes it even better is your enthusiasm is contagious.
Well, thank you, Barry. Yeah. Well, I love this stuff. It's exciting. Of course, I'm so excited that, I worked my whole life, like you said, wrote a ton of papers, right? Wrote a ton of papers and nobody looked at them. Nobody read them. Few people read them.
James, I just want to say congratulations, because your art and your work was appreciated in your lifetime. So I think we're shortening the curve on new thinking and logic. You're still alive, so that's good.
You're absolutely right. I can't complain, can I? I cannot complain. You're absolutely right. You're absolutely. But there still is a lot of work in convincing people that this is important.
Yeah.
So
definitely.
Yeah,
Everybody runs around and says glyphosate, that's the end of the world. But you know what? There's lots more stuff we put in inputs into our land and in our air and water that, it's a problem, glyphosate, but it's not the only one. And clearly, even the things that we would think of fertilizer being a benign thing, it really isn't.
Right. It really isn't. And that's not what plants want.
Yeah.
Plants don't want that. That's not what they do. They don't do that. They don't go find nitrate. They'll absorb it because that's how they get it from bacteria, but and other microbes. But that's not their preferred source. They absorb it because that's what they have those absorb enzymes to absorb it, transporters to transport into the cells. But this is why, because that's what they get from these bacteria, nitrate.
Right. So I want to wrap things up here. Again, thank everybody for attending. Jim, for your great talk. Just have to go do a couple plugs. Terra, we do have some products that can enhance the microbial activity in the soil, add organic matter to it. Just my own property here. I'm in South Jersey, where it's very sandy and typically only get up to about 3% organic matter. With some of the things we've been doing here over a period of 5 or 6 years, we've gotten the organic matter up to 6%, and now we can back off on a lot of the inputs we've been doing. It just maintains itself a very healthy system. So that being said, I want to say next week, well, before I get to that, everybody who attended will get a survey from us. It helps us a lot to get your feedback and where we can go from here. Next week, the speaker is John Ruffini, who has been asking always wonderful questions of you, and he's going to talk about irrigation.
Great questions he had.
Yeah, he's a good guy.
We have one last question. Do we have time for one last question?
Sure.
Yeah, go for it.
Okay. For the industry, do you think landscapers will be unwilling to change because they can make more money by having to continuously fix the issues their practices are causing?
No, I think they can make money in creating more sustainable soils and situations for growing turf and landscape plants. So I think that, in fact, I think that even now, I think, someone told me that, I don't keep tabs of things, but the price of nitrogen is going up. And so people are looking for ways to reduce that nitrogen.
Right. Yep.
Barry. And
yes.
Thank you, Professor Way. Really appreciate this.
You're welcome, John.
As far as changing the industry, is there any organic matter out there that'll soften hard heads? Because I'm telling you, I talk to a lot of these guys, and they are so resistant to any kind of change.
Well,
I
they're
think
not going to live forever.
No, and I think the public will demand change at some point. They won't want a lot of chemicals put on their yards. They may want, it's possible to find ways to manage plants that are highly effective and that work just as good. They may cost a tiny bit more, or they may not. Maybe the price will go down. So, I mean, I know that there are people, there are companies that are doing some pretty innovative and interesting things.
I think if people start replacing fertilizers in general with, humates and mycorrhizals, and
Yeah.
get people to water properly, the homeowners or clients will be a lot happier because they'll be saving their resources and they'll just see much better
Right.
results.
Yeah.
And it's results that really matter.
Yeah. And it's safer for their kids and so forth.
Oh, yeah.
No
their dogs.
runoff.
Right.
You know?
Years ago, Chip Osborne did a study on some of the organic projects he's worked on, and not just the results and everything, but also the economics of it. And what he documented is that at first, yeah, it's more expensive at first, but once you get everything going, there's less maintenance, there's less inputs you have to do. So over a period of time, it's not that expensive.
And there are some, I'll say tricks that people use to fertilize plants that are, like a foliar fertilization approaches to different kinds of crops, where they just put tiny amounts of nutrients into the leaves, and then they get these huge, growth responses in the plants. And so some of these likely are working because they're stimulating these endophytes in leaves of plants. And so a little bit of nutrients, molybdenum and iron, and things that are necessary for nitrogen fixation, put those on the leaves of the plants, and these microbes can get that and fix a lot of nitrogen. And so the plants will grow better. So there's ways to grow plants creatively that people are not using like they could.
I've done that.
Okay. Well, I think it's time to wrap it up. Again, thank you, everyone, and stay tuned for next week's show. Good night, everybody.
Well then, have a good night, everyone.
Good night.
Thank you.
Okay.
Bye-bye.